Okay, so the next speaker is Daniel Jeffries, who's going to tell us about convergence and divergence in the evolution of stickleback sex chromosomes. Please.
Thank you. So, I'm going to talk to you about some work that I've been doing with Katie Peichel in Bern. And most of this work that I'm going to talk to you about, I've done in probably like the last 3 weeks. So it's very preliminary and not very polished, so bear with me a bit. And because of that, I don't have a huge amount of results to show you for this bit. I did that thing where I promised to talk about something and then I still hadn't done the work, and then the work didn't mature as quickly as I'd liked it to have. So I'm going to talk about another project at the end, which I think might be interesting to some or all of you. So we've heard already about some of the sort of bigger questions in sex chromosome evolution. Why do sex chromosomes stop recombining? Why does this process happen more rapidly in some taxa? We haven't heard as much about turnovers today, but also another big question is why do sex chromosomes turn over so often in some taxa and not others? And we've got some great models which we've heard about today, and which could potentially explain some of these characteristics of sex chromosomes that we see. But someone needs to hold these theoreticians to account and actually test these models. So we need some empirical data, of course, to actually refute or support these models. And so Stickleback, I think, are a really nice model nice model system for testing these sorts of things, because they have a really wide range of sex chromosomes, both in terms of the diversity of the sex chromosomes that are used within Stickleback. So you can see at least 5 different chromosomes being used in Stickleback in different species. But also these sex chromosomes range a lot in their age as well. So chromosome 19 is the ancestral sex chromosome in Gasterosteus, in the genus Gasterosteus. So it's at least 10 million years old, but we think that it's much older than that. And then in comparison, we recently discovered the sex chromosome in Brooke Stickleback, and this shows almost no difference between the X and the Y, suggesting that it's very young. But the most work in Stickleback has been focused on this Gasterosteus clade, so I just want to explain to you a bit about what we know about sex chromosomes in Gasterosteus. And this is mostly from work done by Jason Sardell and Mark Kirkpatrick and other members of that lab. Last year it was published, where they basically aligned whole genome resequencing data from black-spotted Japanese and 3-spine back to the 3-spine genome that existed at the time. And they were able to date the various strata that exist on this ancestral sex chromosome. And so we know that stratum 1 on this chromosome existed in the ancestor of these 3 species. But then the sex chromosome lost recombination independently across the 2 lineages here. In 3-spine and Japanese ancestor, it had lost the recombination in stratum 2 and 3. Japanese then fused its Y to the chromosome 9, and then the black-spotted Stickleback independently lost recombination in this region of the chromosome. So this is a really cool system because it allows us to compare how this sex chromosome has responded to becoming sex-linked independently in multiple lineages. But we've also got a sort of almost a benchmark for a region that was sex-linked in the ancestor as well. So a couple of years ago, sorry, there are also extra strata on here, I forgot that. A couple of years ago, Katie and her and several colleagues sequenced the genome of the 3-spine, and that assembly included a phased Y reference chromosome. And this allowed them to infer these strata, these inversions that match up with the strata that we already knew about on this chromosome. And they could also then look at the gene content of this chromosome and show that a huge number of genes have been lost from stratum 1, over 80%, and then some genes have also been lost from the other strata as well, to a lesser extent, reflecting their younger age. And this assembly really afforded a really a great resolution of evolutionary analysis on the Y chromosome of this species. And so what we thought would be good is to add another Y chromosome assembly to this system, so that we could then compare these two Y chromosomes across these independently evolved sex-linked regions on this chromosome. So that's kind of what I'm going to talk to you about today, the process of assembling a Y chromosome, so in the black-spotted stickleback. The three-spine assembly that I just showed you was the result of about 5 years of work from lots of different people and a horrific number of back clone libraries that I've been told about. Our approach was much simpler because we have access to much more advanced sequencing data. So we used PacBio, RS2, not hi-fi reads, Hi-C data, and then importantly, we had access to another data set of whole genome resequencing from males and females from a population close to the population that the male that we're sequencing came from. And this is really crucial to our assembly strategy. So this really is just a summary of that assembly strategy, and the crux of the strategy is essentially we wanted to try to remove reads that come from the Y chromosome before we assemble the genome. And so we did this by using these males and females from our population-level whole genome sequencing dataset to identify male-specific k-mers. So a k-mer is just a segment of a raw read. So by finding male-specific k-mers, we're essentially finding Y-specific k-mers. And we used those k-mers to pull out PacBio reads that presumably are Y-specific, that come from the Y chromosome. We did the same thing again with the Hi-C data as well. So we ended up with a pool of reads for the autosomes and the X chromosome, and then a pool of reads for the Y chromosome. And then we simply assembled those two pools of raw data individually. So that was our approach. We wanted to validate how well this worked. And so the way we did this was first to assemble all of the raw data, just a contig level of assembly of the PacBio data. So this includes reads from the autosomes, the X and the Y. And what I did then was align the sequencing, the whole genome resequencing data from our males and females from our population to that assembly, and just plotted here the female versus the male read depth in small bins along the genome. So this is 1 kilobase. So every dot on this plot is a 1 kilobase bin somewhere in that assembly. And as you would expect, autosomes have roughly equal coverage between males and females. And then we see two reassuring blobs, one with double the coverage in females relative to the males, and then one with about 1N coverage, so haploid coverage in males, and basically 0 coverage in females. So we can be pretty sure that these are our X-specific and our Y-specific parts of the assembly. And so once we knew this, we went through with our read extraction from our raw PacBio data, and then I remade this assembly, and this is my favorite bit of the presentation. This is what we saw, absolutely nothing. And I like doing this. So I think it works really well. And the resulting assembly, so once we had that, these different pools, we went ahead with the rest of the assembly. And the resulting assembly looked a bit like this. This is the autosomes and the X. And I've just compared it here. So this is our black-spotted assembly, all 21 chromosomes, against the three-spine assembly that Katie made, Katie and her colleagues. And we have very nice continuous synteny blocks suggesting that all of our chromosomes are reasonably complete. So we're pretty sure that the X and the autosomes are well assembled. And then our Y chromosome over here looks a bit mad. So this is the Y chromosome assembly from the black-spotted against chromosome 12 and chromosome 19, which are the two X chromosomes that exist in black-spotted because they've had this Y autosome fusion. And I want to compare that with the same sort of synteny plot for the 3' Y chromosome versus the 3' X. And so just to make it clear that because the 3' hasn't had a fusion, this part of this plot is analogous to this part of this plot here, just the chromosome 19 part of the black spotted Y. And so you can see in the 3' that you've got some inversions in here, but you've still got these sort of large synteny blocks where gene order within the inversions is reasonably conserved. In the chromosome 19 part of the black spotted Y, we see no such thing, and things just look completely scrambled. There are a couple of little flecks of maybe a few genes that are in a row in there somewhere. And then on chromosome 12, the chromosome 12 region, which is this neo-sex chromosome region of the Y, we see a lot more sort of retained synteny, except for the oldest stratum on there where synteny is starting to break down here. So in our comparison, we can already say that the black spotted Y looks at stratum 1, which is the, hold on, it's this bit here. Yeah, stratum 1 is the region that's was in the ancestor of both of these species, and even here we've lost synteny. So, so something is going on very differently. This sex chromosome is evolving very differently between these species. And so I haven't had time. I got this assembly about a week ago, so I haven't had time to do much more than this, except to very crudely check gene content for these two Y chromosomes. And so if we ask the question, have the same genes been retained on the Y? Because, of course, these Ys are losing genes over time. The 3' has retained 447 out of the initial 970-ish genes that were on the X chromosome, whereas the black spotted has lost a huge amount more than that. It's clearly lost a lot more genes, but that might reflect the fact that some of the it probably reflects the fact that two of the strata on the black spotted chromosome 19 section of the Y are older than the strata that have evolved on that part of the 3' Y. So these have potentially been losing genes for much longer. But interestingly, of the 123 that are retained in black spotted, 98 of them were also retained in 3' which, and I haven't done any statistics on this, but this to me seems like a non-random proportion of the black spotted genes that have been retained. And that kind of makes sense because we know that genes aren't lost randomly from Y chromosomes. They are lost, genes that are less sensitive to dosage change are lost first, for example. So these should be dosage sensitive genes. So that's something to check. Have the same genes been duplicated on the Y? This is the sort of information that you can only get from a Y assembly. So you can kind of infer gene loss based on coverage against an X chromosome, but you can't infer which genes have been duplicated on the Y or to the Y as easily, at least. 3' have amplified 55 genes that were already on the Y, and whereas the black spotted have amplified only 17, and only 3 of these are shared across both species. And there's the same sort of story when we're talking about genes that have moved from autosomes onto the Y chromosome as well. So black spotted have had fewer duplications from autosomes to the Y chromosome. And actually only one of these genes is the same across both species, and that is the sex-determining gene, which is on the shared stratum. So that's about as far as I've managed to get with it, I'm afraid. Just to summarize, the approach where we're extracting our Y-specific reads seems to have worked really well. So if anyone is thinking of assembling a Y chromosome, I would definitely advise you to think down that line. It seems obvious that the black spotted has undergone a lot more extensive rearrangements and also lost a lot more genes. but it seems to have fewer amplifications and duplications from autosomes. And it's too early, really, to be speculating about what might be driving this, but I can't really help but speculate. And I do wonder if this is some sort of signal for lower efficiency of selection in the black-spotted, where purifying selection may not be as efficient, and so this process of degradation has been more rapid, and also positive selection for amplification of male advantageous genes or duplication from autosomes of male advantageous genes hasn't been as effective either. So I think this might be informed by some population size modelling as well. If we can try to recreate population size into the past, this might be a good next step, at least to put this sort of result into context. So how am I doing for time? Are we good? Okay. So for the next section, then, I just wanted to talk about a pipeline that I've been working on with a master's student for about 6 months, where I was basically trying to solve a problem, and I don't know if anyone else has been in this situation, where you've been working on some species, and you think, okay, it'd be really cool to know what the sister species, what's going on with the sister species sex chromosome, or the outgroup, or something like this. And then you go looking for that species on NCBI just to check that there's any data. And then you find that someone has sequenced a load of samples, and you think, great, this is going to be cool. And then you look for the sex information, and it's just not there. And this is true of so many data sets. I've run into this problem so many times. And I've always thought there must be some way of working some bioinformatic magic to figure out, to try and infer the sex of these things, or try to infer the location of the sex chromosome. And so finally I found some time to try to develop this approach like this. And so you'll have to excuse my cartoons, but this is kind of, I just want to sort of run through the general principle of the approach. So this is the sort of traditional way that we would look for a sex chromosome. So you have some raw read data, you align it to a genome, and then you might compare male versus female read depth. And in your homogametic sex, your read depth shouldn't change along the chromosome, and in a heterogametic sex, you should see a loss of coverage in a region that's been lost from the Y chromosome, for example. And if we were to take any window from the genome and plot the per sample read depth in that window, we would get, in a window where both sexes have 2n coverage, we would find a single modal distribution of sample read depth. But if we were to look at a window in a sex-linked region, then we would see a bimodal distribution of sample read depth in that window. And the same would be true of the same would be true in that data set, regardless of whether we had sex information or not. So if you just remove the color from this, you'd still see a bimodal distribution across your samples. You could also do the same thing with a genotype-based metric like SNP density, for example. Increased heterozygosity in the heterogametic sex would show a bimodal distribution of SNP density in each window across all samples. So regardless of whether you have information for sex of individuals or not, you should actually still have the information about where the sex chromosome is in there. And so our pipeline is essentially just performing a test of bimodality for every window of the genome, given a certain number of samples aligned to a genome. So for every genome in the window, we take a statistic like sample depth or SNP count, or SNP density, if you want to call it SNP density, and we run k-means clustering for that window. So we run k-means for every window independently. And we do that for a value of k from anywhere between 2 to the number of samples that we have. And k-means outputs something called a silhouette score, which is just a statistic that allows you to evaluate how likely that model is relative to other models. And so if we just plot this silhouette score across the genome, we get something like this, just a mess of silhouette scores for different models for different values of K. And what we did was run this on a data set where we know what the answer is, 9 spine, and for the autosomes we get this kind of mess where no value of K is in any way better than any others. And in chromosome 12, which is the sex chromosome, we see K equals 2, which is red here. Sorry, I forgot the key. It has a much higher score, has a much more higher likelihood than any other value. It does seem to drag K equals 3 and K equals 4 with it. I guess that makes sense, because if K equals 2 is well supported, then K equals 3 should also probably be more likely than K equals 17 or whatever. But anyway, you can clearly see that this is this K equals 2 has the highest support here, and that's the only place in this genome. And the other cool thing about k-means is it gives you identities of individuals into the clusters that you've inferred. So if we take the K equals 2 model, then it's clustered individuals into 2 groups, and those groups we can essentially use to infer the sex of the samples that we put in. So we're simultaneously finding the sex-linked region and sexing samples as well. And so this is the k-means result with coverage for this data set. This is the k-means result with SNP density, and you can see there's a couple of individuals where it's got it wrong, so it thinks these are females and not males, or it thinks, I mean, I should say that k-means doesn't know about males and females, it just knows group 1 and group 2. So it just thinks that these are in the other group, basically. But the reason for that is that these 2 individuals are really low coverage, and when you have low coverage, you struggle to call heterozygous SNPs, and when you have lower heterozygosity, you would look like the homogametic sex. So it makes sense that these 2 were wrongly assigned, and so really it's just highlighting how important filtering of data is before putting it in. But otherwise, it got everything spot on. So this is, I think, it's quite promising, and I think it's ready to be rolled out to a lot more systems. The 9-spine sex chromosome is reasonably well differentiated, and so it's kind of easy mode for this pipeline to infer the sex chromosome if it's relatively obvious. You could probably run a PCA on this whole chromosome, and then you'd probably get males and females. But we have tried it on a couple of species which are much less differentiated, and it does still work. The coverage approach isn't useful in species which are less differentiated, but the SNP density approach is much better there. So these 2 approaches are complementary to each other. This pipeline, we initially called it InfoSex. So basically you put genomic data in and you get out sex-linked regions and the sex of samples. And so if anyone thinks they might have a data set that would be useful for testing, or would be willing to test the pipeline and help us figure out bugs and stuff, then it would be super useful for us. And then I've also been talking to Paul recently about turning this into a bit more inclusive tool by including the inference of supergenes in it. And I think people have already done this to some extent, where they've used PCAs in different windows and inferred 3 groups, for example, for the homozygotes, heterozygotes, homozygote alternative. But supergenes are actually, it's almost like a caveat of the InfoSex pipeline, where we might accidentally find supergenes when we're looking for sex chromosomes. So it makes sense to incorporate that into the pipeline so we can distinguish between a sex chromosome and a supergene. The only problem is if there's a supergene with 1 lethal homozygote, then we are going to, and that will look exactly the same as a sex chromosome. But what I will say is that, of course, this is all sort of hypothesis generation exercise, and what we're doing is giving you well, the idea is to predict the sex-linked region and the sex of samples, so that researchers can go out, maybe find a couple of these creatures in the wild, take them back and use a PCR approach, which you can develop from this sequencing data to validate what these pipelines say. And at that point, you will of course know whether it's a sex chromosome or a supergene, based on whether the prediction matches up with the sex. So that's everything. I just want to say thanks to Jan, who's been working on the InfoSec stuff. She was the master's student that we had last year. And then Zhu Yao has been instrumental in the genome assembly of the black-spotted. And then Katie, just for being awesome and paying us all money and stuff. So, yeah, and thanks for listening.